Chapter 14 is Introduction to Host Defense and Innate Immunities. All right. So we're going to go over the host defense mechanism, how we protect ourselves from all these microbes we've been talking about the last eight weeks or so. So innate natural defenses, innate defenses which is present at birth. They're non-specific resistance to an infection. You don't have to learn it. Like we talked about in anatomy, we talk about different types of reflexes, innate reflexes or adaptive reflexes. You don't have to learn it. It's a part of you. Okay? So you have innate reflexes and you have adaptive ones, which you have to acquire to learn. Like when you're driving a car, you see the light turns red, your foot goes from the gas to the brake, or it should anyways. That's an automatic response. You don't have to think about it, just does it. That's a learned response. That's what's called adaptive. So you have innate defenses, which are naturally born with, and then you have adaptive ones, which you've developed over time. That's the difference between them. So the line of you have three lines of defense. The first line of defense is innate, which is it's already in your system, you're born with it. It's non-specific. It blocks everything. Okay. There's an immunological memory associated with it. No, you don't have to learn. You don't have to memorize something, some type of antigen or a receptor you respond to. So actually, let me turn the lights on here too. I'll keep our lights on here. All right. So physical barriers are a part of the first response. The skin, tears, the washing over the eyes, coughing, sneezing, chemical barriers. A low pH. Low pH means high acidity, which kills bacteria. Okay, lysozymes, which dissolve anything. Digestive enzymes. Genetic barriers, like resistance inherited in genetic makeup of the host, where pathogen cannot invade it due to genetic makeup. The second line of defense is also innate. Okay? Mostly non-specific, and there's no memory involved with it. Phagocytosis, inflammation, fever, interferon, and complement. We'll learn all of those as we go in this chapter today. And the third is an acquired response. The third line of defense is acquired. Has to be learned. It's specific for a special type of pathogen. It responds to a certain receptor on that pathogen. So it is due to memory. And these are your T and B lymphocytes and your antibodies. So those are your first, second, and third lines of defense. Okay. So to protect the body against pathogens, your immune system relies on a network of physical barriers. Okay. Immunologically active cells and a variety of chemicals. This is our first, second, and third line of defense. So the first line of defense, okay, is any barrier that blocks invasion at the portal of entry. So like the skin is the first line of defense. It blocks the entry of the pathogen at the portal of entry. And it's non-specific. It blocks everything. Doesn't rely on a specific receptor. The second line of defense, okay, protective cells and fluids, inflammation, phagocytosis. An inflammatory response, got a fever, we've been invading cells in the area. You respond to that pathogen, and it's a non-specific response. And then you have phagocytosis where you dissolve or eat the pathogen. Then the third line of defense, okay, acquired with exposure to foreign substances, produces antibodies and creates memory cells that are specific to that certain pathogen, to that certain microbe that's invading your body. So it's your first, second, and third lines of defense. They don't work separately, they work together. They overlap and redundant in some of their efforts. It's not like a sharp dividing line between the three. Okay. So the first line of defense, our first line, barriers at the portal of entry. So a number of defenses are important parts of the body's anatomy physiology. These are inborn or innate non-specific defenses. We do not have to learn them. They can be divided into three categories: physical or anatomical barriers of the body surface, chemical defenses, and genetic resistance to infection. Okay, certain viruses can't cross different species, genetic resistance. They block the entry of not only microbes, but any foreign agent living or not. Okay. Built-in defenses in the skin. Okay. Outermost layer of the skin, epithelial cells, are cemented together. They're held by the cement between the cells. Okay. They're impregnated with keratin, which gives it the strength. Okay. You get the flushing effect of sweat washing over your skin, keeps it clean. Mucous membranes on the mouth, digestive tube, respiratory tract, etc. Okay. You get blinking and tear production. It goes over the mucous membranes to help protect them. You get saliva that flows to help protect. And the flushing effect of urine, defecation, and vomiting. If something makes you sick, bring it up. Okay. So these are your physical anatomical barriers. You're waxing your ears. Remember, low pH. Low pH means you're raising the acidity. The lower the pH, the higher the acid content. Okay, mucus. Nasal cavity, they can trap pathogens in the lungs. It traps in, and you work with it as you cough up through your lungs. Stomach acid, again, mucus. Normal microbes are in the stomach, help protect it. Normal microbiota. Okay, intact skin, saliva, mucus again up by the nose that traps pathogens. Tears that flow, and sebaceous glands, fatty acids. The oily substance that goes through the hair follicle cells. Okay. So your mucous membranes, mucous coat impedes attachment and the entry of bacteria. Okay. It just basically physically forms a barrier that stops them from entering the body. Ciliary defensive respiratory tree, nasal hairs trap larger particles. So remember the little cilia that are lines of tracheal lung, they all beat in one direction and they beat upwards. So if there's a pathogen gets in your lungs, gets stuck here, those cilia push it up and you clear your throat. All the cilia respiratory beat one specific direction, out of the lungs. Okay, you can see little cilia here that are beating, and those will attach anything and move it up out of the lungs. Okay, non-specific chemical defenses. Okay, from your skin and mucus, sebaceous secretions on the hair follicles. Okay, antimicrobial secretions from special glands. Meibomian glands secrete special antimicrobial secretions. Other defenses are your tears we talked about, saliva, and the skin itself as a physical barrier. Okay, so you have other defenses in the tear, saliva, and skin. You have lysosome, a lysozyme, which is an enzyme that hydrolyzes cell walls of bacteria. Those are your lysosomes. Okay. What they do is they break down the wall of bacterial cells. Defensins is peptides that lyse bacteria and fungi. High lactic acid, okay, electrolyte concentration, okay, in sweat, and the skin's acidic pH. All those act to help defend the body against invading pathogens. Hydrochloric acid in the stomach, again, that dissolves any pathogens in there. It breaks down, it's very nonspecific. It doesn't bind a specific antigen or surface receptor. It just kills whatever's there. Digestive uses, bile of the intestines. Semen contains an antimicrobial chemical, okay, to fight off bacterial infections. The vagina has a very acidic pH, maintained by the normal microbiota of the area. Okay, some hosts are generally immune to the disease of other hosts. Like examples, humans can't acquire distemper from cats, and cats can't get mumps from humans. Most viruses cannot go cross species. Some can, most cannot. Okay. Viruses have a great specificity for their host receptors that bind specifically to receptor on the host cells. That's why they can't go cross species. Some genetic differences in susceptibility exist for other pathogens, differences with members of the same species. Okay. So different, even if they're in the same species, some people can get sick, people don't get sick. Okay. You get some slight difference between members even of the same species. Humans carry a gene or genes for sickle cell anemia. Okay, humans that carry that gene for sickle cell anemia, okay, are called carriers. They're resistant to malaria. Okay. That's why we see a lot of sickle cell disease over in Africa, places where the malaria outbreaks are high. Okay. With the sickle cell anemia, when you're a carrier of it, you don't get malaria. So which of the following is not an example of the immune system's first line of defense? So inflammation, lysosome, hydrochloric acid, sneeze reflex, cilia in the respiratory tract. Which would not be an example of your first line of defense?
Would it be, I don't know, I'm confused between B and C.
B is a lysosome, which means it eats anything.
Maybe C then?
It's hydrochloric acid, it destroys anything. Sneeze reflex is gall and cilia. The only one that's little more specific is inflammation. That'd be a second one, because that's a result of an infection that's entered the skin already.
Oh,
This would be your
it's not
second.
the first. Oh, okay, it's not the first line. Yeah, that makes sense.
Right. Because you have to have something that enters your system first before you get inflammation. So inflammation would be a second line of defense. That makes sense?
Yes, that did.
All right, perfect. Okay, so immunology. Immunology is the study of the body's second and third lines of defenses. This is a primary function of a healthy immune system. Okay. Basically, what an immune system does, or immunology is a study of it, is surveillance of the body. It goes through the body and it just monitors things. It looks for things that don't belong. It's like a surveillance system. It has recognition and differentiation of normal versus foreign material. So the surveillance system has to tell what's normal and what's foreign to be able to respond against it. It attack against and destruction of entities that it's deemed to be foreign. If it figures it's foreign, it's going to destroy it. Now, when things get messed up, like with the HIV virus with AIDS, right, we start to destroy things that are normal because we recognize them as foreign. That's the problem. Okay. So you surveils the body, recognizes normal as foreign when it's healthy, and it attacks and destroys the foreign entities that it finds. Right? White blood cells are your leukocytes. Okay? Erythrocytes are red blood cells, leukocytes are white blood cells. White blood cells have the innate capacity to recognize and differentiate any foreign material. Okay, we have that ability as we're born. We have the ability to do that with our immune system. Non-self, if it's not a self, not recognized as self, it's a foreign material. Self is normal cells of the body. It can determine between the two. Now you have pathogen-associated patterns, which are molecules shared by microorganisms. So those are on the microorganisms, and that's the pattern of molecules that they have on their outside their cell walls. Pathogen recognition receptors are receptors on the white blood cells that recognize these pathogen-associated patterns, these PAMPs. Okay? So all this is being carried out by your white blood cells. Okay, that's your immune system, is your white blood cells. Your red blood cells are determined with exchange of oxygen to keep your body from metabolism. White blood cells is for your immune system. Leukocytes are your white blood cells. Innate capacity to recognize and differentiate any foreign material, non-self and self. Okay, so how do these white blood cells work? These the pathogen-associated patterns, remember those are molecules shared by microorganism, and the PRRs are receptors on the white blood cells for the PMP. So if you look at this is your bloodstream here, okay? So white blood cells can exit the bloodstream and get in the area where the pathogens are. Okay, these are the bacteria right here. These are your pathogens. Okay, and you can see these little bumps on the pathogen. Those are your pattern recognition receptors on the cell here. These pattern recognition receptors recognize the pathogen associated molecular patterns, the PAMPs. Okay, these are the PAMPs on the pathogen right here. Okay, and we have receptors that recognize those on the white blood cells. Those are the pattern recognition receptors on our white blood cells. It recognizes it, and then once it recognizes it as foreign, it starts to destroy it. Okay. Now these receptors, these pathogen recognition receptors, when it finds a normal cell, like right here, receptors find that and it's determined as normal. It does not destroy it. It determines as part of the regular normal cells of the body. Okay, so the pathogen recognition receptors are on the white blood cell, and they bind to either self molecules, which is normal cells, you don't destroy it, or it binds to these pathogen associated patterns, which are on the pathogen, which is on the bacteria. Okay, then it recognizes that as foreign or non-self, and then it destroys it. So the immune system is really large and complex. It's a diffuse network. Okay, and they penetrate cells and fluids. And they penetrate into every organ and tissue of the human body. Body compartments that participate in human function are the reticuloendothelial system. That's all the fibers, connective tissues, all the support structures of the body. The extracellular fluid is all fluid outside the cells. The bloodstream and lymphatic system. Bloodstream and lymphatic carry all the white blood cells to and from where they need to get. Okay? So for an effective immune response, activities in one fluid compartment must be conveyed to the other compartments. They have to all flow together. Okay. So the green here, these are your lymphatic capillaries. This is your extracellular fluid outside the cells. These are your tissue cells here. Okay, and these are the reticular fiber, reticular endothelial system that holds everything together. Okay. So the reticular endothelial system is a network of connective tissue fibers that interconnects other cells and meshes with the connective tissue network of surrounding organs. It's inhabited by phagocytic cells. Those are mononuclear phagocyte system. So in this reticular endothelial system, you have these phagocytes, which go through and monitor and dissolve anything that is recognized as foreign. So these are macrophages. They're ready to attack, okay, and ingest these microbes once it finds them as foreign. Okay. Okay, so once something gets past the first line of defense, then it gets into where we have to recognize it. That's when we get a response by the B cells and T cells of the body, these macrophages. Okay. So here's your macrophages, and it just flows through the system. These are neutrophils, which is a form of white blood cells we talked about a little bit. Dendritic cells is another form that we have in there to help trap bacteria. And you see the reticular fibers, reticular endothelial system that support our body. Okay. These macrophages travel through the systems, all the organs of the body. Okay, so what's blood basically? Because blood is transporting everything. So whole blood, okay, when we talk about whole blood, we're talking about plasma and formed elements. So plasma is the liquid portion of the blood. The formed elements are your white blood cells, your red blood cells, and such. I'm trying to think of platelets, everything else. So of the whole blood, serum is the liquid portion of blood after a clot has formed. So serum is the blood minus the clotting factors. Plasma is 92% water. Okay, that's your liquid portion of the blood. So it's 92% water, and it contains metabolic proteins, globulins, clotting factors, hormones, other chemical acids, gases, which are only function like oxygen, CO2, and so forth. Okay. So this is unclotted whole blood. Unclotted whole blood, you have red blood cells and you have plasma. And you have the buffy coat in between there, separating the two. In clotted whole blood cells, here's your clot. Okay, and on top of that, you have your serum. Serum is everything minus its clotting factors. Plasma minus the clotting factors. Hematopoiesis, okay, or hematopoiesis is the production of blood cells. That's what it means. Hematopoiesis is how blood cells are formed. So it's the production of red blood cells and white blood cells. And it starts from stem cells. Stem cells are undifferentiated cells that are precursors to the new blood cells, whether they're red blood cells, white blood cells. They can be macrophages, dendrites, whatever we form. So you have primary cell types. The primary cell types are your platelets, which are your thrombocytes. Those are your clotting factors. You have your red blood cells that carry your oxygen and your CO2 out of the body. And you have leukocytes. Leukocytes are your white blood cells. Okay? Those are your three primary cells: platelets, red blood cells, white blood cells. Okay, the white blood cells, the leukocytes, are responsible for your immune function, responsible for your immunity. Okay? We have two basic types of categories of different leukocytes. We have granulocytes and agranulocytes. Granulocytes are white blood cells that are thickly granulated. They have multiple granules inside the cells. Agranulocytes are clear. They do not have granulocytes inside the cells. Most of the time, the granulocytes have a lobed nucleus, which could be something that looks like that, different lobes of the nucleus. The agranulocytes are unlobed or rounded nucleus. Okay. So this is how we do it. This is your hematopoiesis. This is your stem cell. Okay. it can branch into two different types once you start differentiating it. You can go to the myeloid stem cell, or it can go to the lymphoid stem cell. The myeloid stem cells for your erythrocytes, which are your red blood cells. It forms your megakaryocytes. Megakaryocytes are these really big cells that kind of look like a snail. And what they do is they branch off little beads here. They branch off little particles, and these are your platelets. Okay, so those form your platelets from this big megakaryocyte. And then you get from the myeloid stem cells, you also got the granular leukocytes. There's 4 granular leukocytes. There's the neutrophils, eosinophils. Here's your neutrophils, eosinophils, basophils, and mast cells. Okay. The mast cells, these guys are what's responsible for your allergies. Those ones that trigger your allergic responses. Now the lymphoid stem cells, okay, those branch off into the agranulocyte leukocytes. And look at the granules inside. Look at the granuloleukocytes that we just talked about, the neutrophils. You see all the little dots inside all these little cells right here? Those are the granules. And look at these right here, down here. Those are your agranulocytes. They're clear on the inside. The granulocytes have lobe nuclei. We just talked about how they kind of oblong shape and kind of different lobes of nucleus. The agranulocytes are rounded, unilobular. Okay, and those are your monocytes, your T lymphocytes, your B lymphocytes, and something called your natural killer cells, NK cells. These NK cells are relatively new development we've learned about. And these guys, remember we talked about how cancer is a disease of the human body, where the normal cells grow at a rapid rate. So it's our disease of normal cells. We don't recognize this form because the cells are normal. Okay? Now, NK cells can recognize those normal cells that are developing faster for some reason. We're not quite sure how they do it, but they can recognize and kill these cancerous tumors. They're surveillance cells that go through the body and they kill off cancer cells. Okay? They have the ability to distinguish between a normal cell that's growing too fast and a normal cell that's not growing too fast. Okay? We can't do that. Our chemotherapy wipes out everything. But NK cells specialize in function in going after certain types of viruses and cancer cells. And then from there, we have different cells that develop. We have plasma cells. Okay, produce large quantity of antibodies. We have dendritic cells. This come off of your from monocytes. We can have your macrophages and dendritic cells. Dendritic cells from your nerve cells, and they can tend to trap other things for the phagocytic cells to dissolve them. So there's basically how the hemopoietic system works. From our main stem cells, hemopoiesis, branching into 2 different ones, lymphoid and myeloid, and the different divisions that we have for those. Okay, so we talked about the different leukocytes. Okay, we have neutrophils, 55 to 90%. Okay, make up for your lymphocytes. Oh, okay, these are lobe nuclei with lavender granules. Okay, phagocytosis, they eat other cells. Eosinophils make up 1 to 3% of the white blood cells. They're orange granules, they're bilobe nuclei, and they destroy eukaryotic pathogens. Basophils make about 0.5%. They have constricted nuclei, they have dark blue granules. They release chemical mediators, potent chemical mediators that destroy pathogens. And from the base, we have mast cells, which are non-mobile elements found in connective tissue. Okay, those are from the slide before. This is your neutrophil. You can see the this is one of the granulocytes. This is an eosinophil. Again, see the lobe nuclei, and you see all the granules inside of here. And the basophil, same way, lobe nuclei, and all those granules. Agranulocytes, ones without granulocytes, about 20 to 35% of the lymphocytes make up are made up of agranulocytes. And these are the ones that give you a specific immune response. Okay. B cells, human immunity, activated B cells produce antibodies. They make the antibodies. T cells are some immunity. Activated T cells modulate the immune functions and kill foreign cells. Okay, so the B cells produce these antibodies. The antibodies tag the cells as foreign Once their attack is formed, the T cells kill them. So you have monocytes, macrophages, about 3 to 7%. The largest of the white blood cells, okay, are these monocytes, which are your macrophages. Once they go around and eat, destroy, dissolve the foreign pathogens. Okay, macrophages for the final differentiation of monocytes. And dendritic cells. These cells have all those little branches, like in the nerve cells, and they tend to trap pathogens and have participated in the immune reaction. Okay. So these are your lymphocytes, white blood cells. These are the agranulocytes. Yeah, it's one nucleus. It's not lobed and it's clear. The cytoplasm here is clear. There's no granules inside that cytoplasm. This is a monocyte. It's going to develop again. You see it's one nucleus and there's no granules inside the cytoplasm. So some of we just talked about. We have neutrophils, 55, 90%, phagocytosis. Okay. Lifespan of 2 days with only 4 to 10 hours spent in the circulation. Multi-lobes, small granules, contain digestive enzymes. Eosinophils, 1 to 3% of the white blood cells. These are used for the destruction like parasitic worms. Okay. Mediators of our allergic responses, found a much higher number in the spleen and in the bone marrow. They're bilobed, okay, multilobular, large orange granules, contain toxic proteins, inflammatory anti-digestive enzymes, and the basophils. Okay, paying about 0.5% of the white blood cells. Okay, these are what's active in your allergies, inflammation, parasitic reactions, allergy inflammation. These are cytoplasmic granules, contain histamine and prostaglandins. That's your histamine response from allergy. Okay. They're pale staining, constricted nuclei with dark blue, black granules. Monocytes. Monocytes are 3 to 7% of your white blood cells, and those are phagocytosis. Okay. Turned into macrophages and dendritic cells is what it finally differentiates into. Monocytes secrete several chemicals that monitor the functions of their immune system. It is the largest of the white blood cells. So the largest white blood cells are your phagocytes, which are your monocytes. That's where they came from. Nuclei are very large, they're oval shaped, and often no cytoplasmic granules. They're agranulocytes. Okay. Lymphocytes. Lymphocytes make up 20 to 30% of your white blood cells. Okay. These are specific acquired immunity. Those are your B cells, your T cells, your NK cells. Okay. 2 types exist: T cells, responsible for some immunity, and B cells, responsible for making antibodies, humoral immunity. They're small spherical cells with uniform staining, dark round nuclei. So the T cells, the B cells, recognize it as foreign, makes the antibodies, and they mark these foreign bodies as non-self. The T cells find these markers that have been put on there, and then they eat or destroy those existing cells that are non-self. Okay, I mean, the immune system is very, very complex. This is just hitting the basic of it. Okay. So erythrocytes. Erythrocytes are your red blood cells now. Okay. Those develop along a different line. The erythrocytes develop in bone marrow. Okay, stem cells lose nucleus and become biconcave sacs of hemoglobin. So as the red blood cells develop, they develop in your bone marrow. But you got to remember the red blood cells and then the B cells and T cells all develop in the bone marrow. They don't become active. The B cells become active in bone marrow, that's why they're B cells. The T cells become active in the thymus. Okay, but they all start off in the bone marrow. So the erythrocytes lose their nucleus. So there's no red blood cells have no nucleus, they don't need one. They only live about 120 days roughly. Okay, and all they are biconcaved. So concave on both, I'm not very good on a disc, but concave's in the center, and it's just filled with hemoglobin to absorb oxygen and carbon dioxide. Okay. So those are your erythrocytes, your red blood cells. Okay. Platelets form donors in circulating blood that are not whole cells. Platelets are formed that broke off the megakaryocytes, those little things that help with clotting. The lymphatic system provides an auxiliary route for extracellular fluid to the circulatory system. The lymphatic system drains back into the bloodstream. I think the left subclavian vein that comes back in. Acts as a drainage system for inflammatory responses, renders surveillance, recognition, and protection against foreign materials. It's part of our immune system. The lymphatic system runs through the entire body. Okay, drains back into the right subclavian vein. Okay, back into the bloodstream when it's gone through the lymphatic system. Lymphatics is a detailed roadmap through the body. This is lymphatics of the breast. These are your axillary lymphatic nodes, which is under your armpits. But you can see on the diagram here that they have you see in the bone marrow where all these start to develop. These are all lymphatic vessels run through the entire body. You have the tonsils, which are your MALT systems. Okay, you have the thymus is where your T cells develop. The T cells become active. Okay. They become activated so they can start their function. MALT, which is your mucus-associated lymph tissue, which is in the breast. And the spleen, which actually recycles your red blood cells. And you have the Pierce patches of your GALT. Okay, and then the inguinal lymph nodes in the groin area. Okay, so lymphatic fluid, it's separate than the blood vessels. Okay, lymph is a plasma-like liquid. It's carried by the lymphatic circulation. It's a different circulatory system than the blood circulatory system. Okay, it drains back into the blood circulatory system and then gets recycled. It's formed when blood components move out of blood vessels into the extracellular spaces between the cells. It's made up of water, dissolved salts, and about 2 to 5% proteins. It transports white blood cells, fats, cellular debris, and infectious agents. It does not transport red blood cells. Lymphatic capillaries permeate all parts of your body except the central nervous system, the bone, placenta, and the thymus. Those are the only exceptions to where the lymphatic system does not innervate or permeate. Thin walls in the lymphatic system and lymphatic vessels. Okay, it's easily permeated by extracellular fluid, then moves through the contractions of skeletal muscles. That's how what's the fourth. There is no heartbeat for the lymphatic system. It just uses the force of skeletal muscle contraction to move through the body. Functions to return lymph through circulation, flows one directional towards the heart, eventually returning to the bloodstream. So lymph only flows in one direction, just like our blood circulatory system only flows in one direction. So you see the lymphatic system and our cardiovascular system. Cardiovascular system controls our blood, flows through the heart, goes through capillaries in the lungs, flows back in the heart and gets pumped through the rest of the body. Lymphatic system is a totally isolated system from that, throws the entire body and flows back up and back into the heart to the bloodstream. Okay. So that what shows for lymphatic circulation, the finest level of lymphatic circulation begins with blind capillaries. Okay, they pick up foreign material from surrounding tissue and transport it in the lymph away from transport in the lymph, away from the extremities, right, through the system of small ducts. So it's just like in our capillary system, these are small lymphatic ducts, and they transport the foreign material away from the affected site. These ducts carry lymph into a circuit of larger ducts, okay, that ultimately flow into clusters of filtering organs called your lymph nodes. You have lymph nodes, your cervical lymph nodes, one's under your mandible here. Those ones get inflamed during sicknesses, illnesses. Okay. So a section through a lymph node reveals different afferent ducts. Afferent means incoming. Where's our efferent? And efferent means outgoing. So afferent ducts come into the lymph node. Efferent ducts come out of the lymph node. Okay, so the afferent ducts drain lymph into the sinuses. That's just these cavities into the lymph nodes that house several types of white blood cells. Here, foreign material is filtered out and processed by these lymphocytes, macrophages, and dendritic cells to see if it's self or foreign. Okay, lymph continues to trickle from the lymph nodes through efferent ducts, which is outgoing from the lymph node once it's passed through the lymph node, into a system of larger drainage vessels, which ultimately connect with large veins near the heart. In this way, cells and products of immunity continually enter the regular circulation as it goes through the body. So here's your lymphatic system. Here's your lymphatic capillaries. Those are very porous. They foreign materials gets into those. They flow into this is your afferent lymphatic ducts. This is your lymph node. This is what a section of lymph node looks like. You can see the sinuses inside it, where it flows. Then it flows out of the lymph node. This is your efferent lymphatic ducts. Okay. And it feeds into larger and larger ducts as it goes. Okay. Allows it to flow back into the general blood circulation. Okay. So we have primary organs. The primary organs are the site of lymphatic origin, okay, and maturation, where they grow. The thymus gland, like I said, the thymus gland is where the T cells mature. Bone marrow is where B cells mature. Okay. So both the T cells and B cells are formed in the bone marrow. The B cells become activated in the bone marrow, where they're at. The T cells have to travel to the thymus to become activated. Okay. Then you have secondary organs. Secondary organs are your circulatory systems. Okay. You have your lymph node system and your spleen. Lymph nodes drain infected materials from the extracellular fluid through afferent lymphatic ducts into the lymph node, where it goes into sinuses, where all these white blood cells are. We identify it itself, we let it go back through, or identify it as foreign, we destroy it. And then it goes through the efferent nodes back into the circulatory system. The spleen, as it goes through the circulatory system, is basically recycles red blood cells, takes the red blood cells that are damaged, destroys them, and reuses the parts to make new red blood cells. Okay, collection of cells distributed through the skin and mucous membrane. We have different groupings of these lymph nodes. We have the MALT system, which is mucosal associated lymphoid tissue around the breasts. We have the SALT, which is skin associated lymphoid tissues. And we have the GALT, which is gut associated lymphoid tissues. Okay. Again, where are pathogens going to attack? They're going to come in the skin. We're going to our lymphatic tissue there. And through we're going to eat them when we eat bacteria. Okay, those are gut associated lymphoid tissues to help filter out the stuff coming in through our digestive system. All right, so we have the thymus we talked about, lymph nodes we talked about, and the spleen. So the thymus has very high rate of growth until puberty. After puberty, it begins to shrink. The thymus is the site of T cell maturation. That's where T cells mature. They are not produced there. T cells are produced where? They're produced in the bone marrow, just like B cells. So the lymph nodes are very small, encapsulated bean-shaped organs. They're along lymphatic channels. So the afferent lymphatic vessels drain the fluid in. The efferent lymphatic vessels take the fluid out of the lymph nodes. Okay. They go to the larger blood vessels of the thoracic and abdominal cavities. And the spleen is between your diaphragm and the left of the stomach. It's similar to a big lymph node. It's filled with circulating blood cells, removes the worn out red blood cells and other pathogens. It's a function of the spleen. And remember the thymus gland, located roughly there. Okay, there's a medulla and a cortex like all other glands. Cortex is the outer layer, medulla is the inner layer. Okay, and the thymus gland is where the T cells go to mature and become active. Okay, so where do, okay, we should get this one right. So where do T cells mature?
They mature in the thymus.
Thymus, perfect. They mature in the thymus. They're produced in the red blood cell, the bone marrow, but they actually mature in the, so we have talked about our first line of defense, and now this is our second line of defense. Remember, the first line of defense is just stops it from getting into the body. Your skin, your mucous membranes. It just prevents it from getting into the body, your pH system, acidic pH. Now, once it gets in the body, then you get your second line of defense. That's inflammation. This is still a general response. It's not specific to a certain type of bacteria or certain type of virus. You get inflammation in response to anything that comes in. So these are mechanisms that play an important role in our host defenses. You have recognition, you have inflammation, you have phagocytosis, you have interferon, and you have complement. And we'll describe each one of those as we go through this. Okay, so they're still non-specific. They're not binding to any receptors yet in their effect, but they support and interact with specific immune responses. Remember, we talked about there's not a line that separates first from second from third. It's all kind of a gray area, and it all kind of overlaps in our defense systems. They all kind of work together in cooperation with one another. So the inflammatory response, this is a reaction to any traumatic event in our tissues, and we attempt to restore homeostasis. Homeostasis is our normal body equilibrium, controlled by our hypothalamus, that we continue to maintain our blood pressure, our body temperature. We tend to maintain all things to keep our body in a state of what's called homeostasis. It helps to clear away invading microbes and cellular debris left by the immune reaction. Because we have the immune reactions, we dissolve, these macrophages eat these microbes, right? They got to go somewhere. We got pieces and parts left over. Okay, so we got to clear those out. Okay, that's your cellular debris. The inflammatory response is several factors. Redness. When we get inflammation, we get redness. Okay. It increased circulation and vasodilation in response to chemical mediators. Okay, so the bloodstream comes there, and you got vasodilation, so the blood vessels dilate in that area, so more things can respond to that area. That makes it red with increased blood flow. We get warmth, heat is given off by the increased blood flow of the area. We get swelling, increased fluid in the body, okay, increased fluid in our tissues as the blood vessels dilate. Edema, so the water or the liquid blood leaks out and it starts to swell up. White blood cells, microbes, debris, fluid collect, and it forms pus, that prevents the spread of infection. So the pus forms a pocket around it, and it keeps the infection controlled in that area. It doesn't allow these bacteria to keep going everywhere because it coats them in this pus pocket that keeps them there. And then we get the all famous pain, stimulation of these nerve endings from this increased pressure. We get increased water in the area, increased pressure on these nerve endings. They're going to fire, and that's where we get pain. And these are all things we know. You get inflammation, it gets redness, it gets warm, we start to swell up, and it hurts. Okay, and these are the reasons why. And you can see the inflammatory response. Okay, so we get a major event, we get a trauma. This is the skin, this is a needle, punctures the skin, we get a hole. So bacteria go in the wound, they enter that hole. Now we got a hole in the skin, bacteria can enter the body. This is our first line of defense. Now we got a break in it. Okay, so once we get this break here, these mast cells under the epithelium release certain chemical mediators. Okay? And we get vasoconstriction here. So we shut this area down. We stop the blood in that area and we start to shrink it down. So here's our injury, the immediate reaction. Blood vessels narrow, vasoconstriction to form the blood clots. Mast cells release chemokines and cytokines into the injured area. That's your initial immediate response right there. Then vascular reactions, nearby blood vessels dilate. Okay, so now we start to dilate blood to bring more blood into the area. You get increased blood flow, increased vascular permeability. More of the stuff leaks out of the bloodstream. Increased leakage of fluid forms this exudate, forms your pus. So here's your clot starting to form on the outside of the skin. Then the bacteria that are now invaded, bacteria have got past our first line of defense. Neutrophils, those are leukocytes. Okay. You get seepage of plasma and migration of white blood cells out of the blood vessels. So now the white blood cells, these are white blood cells right here, they start to leak out of the blood vessels so they can invade that area. Okay. The edema and pus formation. Edema just means swelling from the extra water. You get collection of fluids, edema, swelling, infiltration by neutrophils, and we get the formation of pus that seals the area. So here's your scab. These are the neutrophils invading the area. They're coming out of the blood vessels. This is the pus pocket that's starting to form the exudate. And here's the fibrous exudate that's starting to wall off this area. Okay, you get your scar formation, which is your resolution. So macrophages, lymphocytes, and fibroblasts migrate in. Okay, get this innate immune response, and this can be learned to repair of injury, scar, and loss of normal tissue. So here's your scar. These are your lymphocytes, your fibroblasts to help now increase that area. Okay. And then our macrophages, which are large neutrophils, which are starting to absorb or eat, consume the bacteria. Okay. And this is a reticular formation. This is a reticular scar formation that forms around an injury through the red blood cells. That's a hole in the skin. This is reticular formation that forms this network to help form the blood clot. Okay. So leukocytes are white blood cells. They're a response to an inflammation. Okay. And what we have is, this is when the neutrophils start to release it, they release a chemotactic factor. These are chemicals that form, and it forms a gradient. So these neutrophils follow this gradient towards the invading pathogen. So you get, in other words, if the pathogens are here, the leukocytes form all these little chemotactic factors, and it gets less and less as it goes out here. Okay? So the neutrophils come out of the red blood, out of the bloodstream, and they find this gradient, and they travels, like following a scent. They follow to where the largest collection of chemotactic follow. That's called chemotaxis. These are the neutrophils following basically a scent. It's following down a concentration gradient. It starts small, but they go to where the concentration is the greatest. It pulls the neutrophils there. So it pulls all the white blood cells that we want into the area that we needed to fight the infection. Okay, so those are the two terms you need to be familiar with, especially for your nursing exams. Diapedesis. Diapedesis is these white blood cells that can come out of the blood vessels into the tissue. Okay, it's migration of cells out of the bloodstream into the tissues, and they're white blood cells. And chemotaxis is where they go. It means you're going to follow a chemical gradient that's being set up by release of chemotactic factors from the neutrophils that are already there from their initial response. Okay, so these neutrophils come out of the blood cells, so you need to know diapedesis and chemotaxis. And that gets our white blood cells to the area where the infection is. Okay. So in conjunction with inflammation, okay, we get circulating pyrogens. Okay. These pyrogens reset the hypothalamus. Remember, the hypothalamus creates a homeostasis, allow the body to maintain a certain body temperature, pH, etc. These pyrogens change the thermostat. They change the hypothalamus, okay, to increase your body temperature. That's why a lot of times with infections you get a fever, because these pyrogens release trigger the thermostat, which is the hypothalamus, and say, we're cranking our body temperature up. This does a couple of things. This signals muscles to increase heat production, and it causes vasoconstriction, because now you got a lot of heat, you're going to constrict the body. This is going to keep the infection confined. Okay, it's also going to increase the heat, which kills a lot of bacteria. So it's going to do a couple of things when you increase your thermostat, your hypothalamus, to increase your body temperature. And again, it signals muscles to increase heat production, and you get vasoconstriction. So you get exogenous pyrogens, are products of the infectious agent. Those are your endotoxins. Those are released by the bacteria, by the microbe that's invading your body. Or you get endogenous pyrogens. Okay? Those are liberated by our own cells, by the monocytes, neutrophils, macrophages during phagocytosis. This is interleukin-1 and tumor neural necrosis factor, TNF. Okay? So exogenous pyrogens are released by the bacteria. Endogenous pyrogens are released by our own cells. And again, they affect the hypothalamus, which is your thermostat, to increase body temperature. Benefits of the fever. We talked about why crank the thermostat up. Inhibits multiplication of temperature-sensitive microorganisms. Okay, it slows down the growth of the bacteria, pathogen in the body. Impedes nutrition of the bacteria. Reduces available iron. The bacteria can't feed as readily. And increase metabolism and stimulation of immune reactions and protective physiological processes. Okay. So this is all, we're still talking about inflammation. This is inflammation is the second line of defense. Along with inflammation, we get phagocytosis because we brought in our neutrophils, we brought in our macrophages. The macrophages have followed that neuropoiesis, left the bloodstream, followed the chemicals down, chemotaxis to the area. Now we got macrophages in the area. And the macrophages are phagocytic cells. Okay, so cervic tissue compartments discover microbes. They discover the microbes, particulate matter, and dead or injured cell. They find them. Then they ingest them and they eliminate them. They extract immunologic information for matter. So they take the pieces they've eaten and say, look, these are things that you need to recognize as being bad for our body. They're not part of our own body. So then we can start our third line of defense. We can start our memory. We can start our immunological response to that. So major category are phagocytes, are neutrophils, eosinophils, and macrophages. The neutrophils are a general purpose one. Okay, they react early to bacteria, for materials, and to damaged tissue. Eosinophils are attracted to the sites of parasites or parasitic infections and antigen antibody reactions, B cells and T cells. The macrophages are the big ones. The largest ones are derived from monocytes. They're scavengers, and they process foreign substances together for reactions with B and T lymphocytes. They're the ones that do most of the dissolving of the pathogens. So after leaving the bloodstream and entering the tissue, monocytes are transformed by inflammatory mediators into macrophages and dendritic cells. Macrophages, again, absorb pathogens and destroy them. The dendritic cells, like the dendrites of a nerve cell, form a network and help trap the pathogens in that area. So macrophages and dendritic cells can remain nomadic or take up a specific organ. So they can flow around the body, keep going, or they can take a specific site and work in that area. Again, here's your inflammatory response. Okay. So phagocytic recognition, this is what we talked about. Okay. This is a macrophage, and we have these little receptors on this macrophage. And when the macrophage finds, remember the PMPs we talked about? Those are the molecules on the surface of the bacteria. It finds those receptors and it binds them. It pulls it together and says, and it triggers this. Now we get a response. This responds to different cytokines here, interleukins, and we get inflammatory mediators responding. Okay, so it activates our macrophages, and you start to destroy it. So protein receptors with cell membranes or macrophages, like the toll-like receptors, detects foreign molecules, signals the macrophages produce chemicals to stimulate an immune response. Okay. Remember chemotaxis? Chemotaxis is the release of chemical gradient, so macrophages can follow it to the site of infection. Okay, so you get chemotaxis, you bring it in there, you get binding to it by recognition of those receptors, and then you get ingestion where it destroys it. So phagocytes migrate to inflammation, following a gradient of stimulant products. That's your chemotaxis. Then they use the TLRs, binding pathogens associated with like PAMPs, receptors to bind to those areas, to the bacteria, and then start to ingest them. Okay. Phagocytes extend pseudopods. Pseudopods are an extension of your cell membrane. So if you're binding to it, they can extend the cell membrane like that, so you can get an engulfing of these bacteria. So phagocytes extend pseudopods and enclose pathogens in a vacuole called a phagosome. In a short time, the lysosomes with antimicrobial substances fuse with the phagosome to form a phagolysosome. Okay, so this is your phagocyte and a lysosome fuse, so you get digestive enzymes, and that digests the pathogen. And we'll show you a picture of that too in a second here. So we get destruction and elimination of the invading microbe. Okay, in an oxygen-dependent system, we get what's called respiratory burst, liberation of lactic acid, which is going to lower the pH. Okay, lysosome and nitric oxide, which are all toxic. Undigestible debris is released from the macrophages by exocytosis. Exocytosis, remember we talked about exocytosis is when you have your cell, and then it forms a little pocket inside, and then all these stuff gets kicked back out as waste as the cell forms back to its original shape. Okay. So chemotaxis, by the phagocyte, we leave the bloodstream. Here's your bloodstream. We leave the bloodstream. We follow the chemical gradient set up to the highest concentration, and we find bacteria. Lo and behold, there's all the bacteria. Okay. Now it starts to bind this bacteria cells. Here's the bacteria. These are bacteria, and this is a cell wall binding to it and starting to engulf it. Okay, so we get adhesion of bacteria. We get those binding to our receptors. Now we start to get the cell membrane. This is our human cells folding around the bacteria here. Okay, and then it folds it into a vacuole. This vacuole fuses with a lysosome. This is a lysosome comes down. This is lysosome production. The lysosome and the vacuole fuse. Now you got a vacuole with the digestive enzymes inside. what do diazoxyribonucleic acids do? They break down or destroy the bacteria. And then they get this debris that's left over, the bacteria are killed. And then exocytosis, it kicks out that waste to go out to urine or feces and get rid of it in the body. And that's basically how it works. That's just an overview of it. Now, interferon. Interferon is one of our cancer treatments. Okay? Small protein produced by certain white blood cells and tissue cells. There's three major types of interferons. There's interferon alpha, interferon beta, and interferon gamma. Okay, the interferon alpha is a product of lymphocytes and macrophages. The interferon beta is a product of your fibroblasts and epithelial cells. And the interferon gamma is a product of your T cells. Remember, the T cells are the immune cells that recognize the marked cells that are marked as non-self and destroy them. They're all produced in response to viruses like cancer, RNA, immune products, and antigens. Produce those three types of interferon. See, all three types bind to the cell surfaces and induce expressions of antiviral proteins. Antiviral proteins are proteins that are going to help destroy the virus that are invading the body. Okay. They inhibit expression of certain cancer genes. The interferons alpha and beta stimulate the phagocytes to start eating material. Interferon gamma is a regulator for the macrophages and the T and B cells. Okay. So here's your antiviral activity of interferon. This is your viral infection, comes in. Here's your virus, and it puts the virus together and makes new viruses. That's how it works. Now, the viruses are marked. The interferon gene is here. You get synthesis of interferon. This interferon transfers to the next cell through these receptors, and this signals the activation of certain genes. These genes come up here and stop the assembly of the viruses. So it blocks virus replication, and no viruses leave the next cell. That's how interferon works. Complement. Complement was the other portion. Consists of 26 blood proteins that work in concert to destroy bacteria and viruses. Okay. Complement proteins are activated by cleavage, which is a cascade reaction pathway. And the show goes through 1, 2, 3, 4, etc. as it goes down through the pathways. Okay. There's classical, there's a lectin pathway, and there's an alternative pathway. Classical is activated by the presence of antibodies bound to microorganism. Lectin is a non-specific reaction of a host serum protein that binds mannan. Okay, binds to it and kicks a pathway that starts its cascade going. Alternative begins when a complement protein binds to a normal cell wall and surface components of microorganisms, and that triggers a cascade. Okay, and here's your different class. This is a classical pathway, this is a lectin pathway, and alternative pathway. And basically, they're all going to accomplish the same thing. They're all going to accomplish C3 convertase converts the C3 molecule to the activator of the cascade C3. And once you get C3, this is your activator, it forms down that cascade, and it forms these little spikes that form into the membrane. And you can see these little spikes that come together, form a hole, and it's going to blow a hole in the membranes of cell wall and membranes of bacteria cells. So all three pathways are all going to end up forming C3 as your end result. Different factors are going to lead to that, different ways of doing it, but they're all going to have the net result to form that complement cascade. Okay, so the cascade application, you get C5 factors activated by C3b, converts to C5b, C5b becomes bound to the membrane, serves as a starting molecule for the chain of events that assemble a complex in C and D, part C and D of this one. Then you get polymerization of C5b, reactive site for the final assembly of an attack complex. Okay, then you get C6, C7, and C8 aggregate, become integrated into the membrane. They form a substrate, which is the final component C9. Okay, they can bind up to 15 of these C9 units. Okay, go in the central core, and they form an attack complex called an MAC, the membrane attack complex. Okay, these produce hundreds of tiny holes in the cell membrane, causing cell lysis and death of eukaryotic cells and many gram-negative bacteria. This is what they do. They form these spikes. And as these spikes come together, they group into this ring and they form holes in the bacterial cell wall membrane. And these little holes in the cell wall and membrane what these do is allow the cell components to leak out and basically kill the bacterial cell. Okay. So two products of this cascade reaction, as you see there, C3 and C5, have additional inflammatory functions. Both these molecules stimulate the mast cell degranulation, enhance chemotaxis of white blood cells, and act as inflammatory mediators. Okay, so they enhance this gradient that's set to draw more to the area so we can kill more of the bacteria. You see, as these hundreds of holes invade this bacterial cell, you can tell the cell wall is breaking down and everything leaks out and the cell dies. Okay, so an overview of our major host defenses. Okay, innate is non-specific. Adaptive is specific. So innate is non-specific. It's our first line defense. Physical barriers, chemical barriers, and genetic barriers. First line of defense is a surface protection composed of anatomical and physiological barriers that keep bacteria from penetrating sterile body compartments. That's the definition of it. Second line of defense is your inflammation response. This can only occur after it gets through the first line of defense. Then you get inflammation. You get the interference we talked about going through a cascade. You get the phagocytosis and you get the complement system. Second line of defense is a cellular and chemical system that comes immediately into play if the infection agent makes it past the surface defenses. Okay, it's got to get past the surface defenses first, and then we get the inflammatory response. We get our secondary line of defense. Examples include phagocytes that destroy foreign matter, and inflammation, which holds infections in check. It keeps it in a certain area. It encapsulates it. Acquired, a specific response. This is our learned or a memory response. B and T lymphocytes, antibodies, and cytotoxicity. So the third line of defense, okay, includes specific host defenses that must develop uniquely for each microbe, okay, through the action of specialized white blood cells. We have to mark each microbe as foreign and learn that as a foreign material. Okay, this is a learned response. This form of immunity is marked by its activity towards specific pathogens and development of memory. These B cells and T cells learn what is foreign. That's how vaccines work. We give an injection into our bodies as something that's foreign. We build up these antibodies to it. We give them doses low enough or an inactive form of the virus, attenuated, so it can't cause the disease, but it still releases the pathogen, so we can develop immunity to it. So now when we're exposed to the real thing, we have the antibodies in our system and they kill and attack it before it can take hold. So which of the following defenses is most likely to be active during a viral infection? Which one of these fights viral infections? Which one of these produces the antiviral genes to become active? Remember, if we go back and look, antiviral genes right here, antiviral proteins produce our antiviral type. So, okay, so which of the following active during a viral infection is interferon.